All right, thanks for the invitation. My name is Jeihun Kuack, and I'm a ORI's postdoctoral fellow at the Division of Pediatric and Maternal Health, FDA. And my presentation is about prediction of maternal pharmacokinetic in pregnancy using physiologically-based pharmacokinetic modeling. There is no conflict of interest, and the views expressed in this presentation do not necessarily reflect the official views of the FDA. So some backgrounds about the drug. Bictegravir is integrase transfer inhibitor widely used as part of 3-drug combination regimen for the treatment of HIV in adults and pediatric patients who have no previous antiretroviral treatment history or to replace the current regimen in those who are biologically suppressed on a stable regimen. The drug is primarily metabolized by cytochrome P450-384 and UDP glucoronolacyltransferase 1A1, and the drug is also highly protein-bound, mainly to serum albumin, more than 99%. The pregnancy has been observed to elevate the metabolism mediated by CYP3A4 and UGT1A1. Additionally, there is a reduction in protein binding property during the gestational period. Those physiological changes during pregnancy may impact the pharmacokinetic, including Cmax and AUC. Thus, the therapeutic dosing in pregnant patients living with HIV may be impacted by those changes. So we conducted the study to predict the PK of Bictegravir in pregnancy using PBPK modeling and evaluate model performance with observed PK data from the International Maternal Pediatric Adolescent AIDS clinical trial, abbreviated as IMPACT 2026 study. So the picture briefly shows how the pregnant PBPK model was developed. So first of all, the model was built using in vitro and in vivo data. Then the model underwent validation process in non-pregnant population, and the model went through some optimization process until it successfully predicted PK parameters of non-pregnant population. Gestational age-dependent physiological parameters were incorporated as system parameter in SimCip, and the model then underwent validation process in pregnant population again until it passes the performance testing. So as it was described before, the Bictegravir PPK model in healthy non-pregnant virtual participant was constructed in SimCip version 22, and drug-specific absorption, distribution, metabolism, elimination parameters, and physiochemical parameters were incorporated along with system parameters. The model then went through the external validation steps with clinically observed PK data from clinical study in healthy non-pregnant participants. On the right side of the screen, the graph shows plasma concentration versus time profile of 50 mg single dose of Bictegravir in healthy non-pregnant participants. The solid line and shaded area represent geometric mean of predicted concentration versus time with 95% confidence interval, and the black dot represents geometric mean of clinically observed data from Solovic study in control arm. The model then underwent further validation process with 2 different drug-drug interaction scenarios with adagenabir and voriconazole to validate the performance of the model to predict CYP3A4 and UGT1A1 mediated contribution in metabolism. Adagenabir is a potent UGT1A1 and CYP3A4 inhibitor, and voriconazole is CYP3A4 inhibitor. Clinical PK data were taken from FDA Clinical Pharmacology NDA review. The standard acceptable range is 90% confidence interval for the geometric mean ratio PK parameter within 0.8 to 1.25. In both scenarios, forward errors of AUC and Cmax are highlighted in red colors. And using those 2 drug-drug interaction scenarios, the Bictegravir model successfully passed the validation process in healthy participants. After it passes the external validation processes, we were confident in performance of the model in non-pregnant population, and the model was then extended to pregnant population in 2nd and 3rd trimester. The previous study by our team on changes in laboratory parameters during pregnancy discovered that there are some changes in albumin, alpha-1 acid glycoprotein, and hematocrit levels in pregnant patients living with HIV. The differently expressed protein binding property and hematocrit in in people living with HIV in second and third trimester of gestational age were expressed in polynomial equations, and the developed polynomial equations were incorporated as system parameter in SimSip. And protein binding levels of people living with HIV during pregnancy were adopted from IMPACT 126S arm, and prediction in pregnancy were compared with clinical data from IMPACT 2026 arm. So the graph shows the predicted plasma concentration versus time profile of virtual patient in non-pregnant, and second and third trimester. The black solid line shows the concentration versus time curve in non-pregnant virtual patient, and blue and red line represent simulated concentration versus time curve of virtual patient in second and third trimester. The reduction in Cmax and AUC was observed using the PBPK model as is shown in the graph. And then we compared our prediction to the clinical data from IMPACT 2026 study presented at Conference on Retroviruses and Opportunistic Infection 2023. The solid line represents predicted concentration of Bictegravir over time in geometric mean, while the dotted line represents clinical data from IMPACT in median value. We found that both simulation and clinical trials show consistency in finding reduction of drug exposure during second and third trimester. And here are some numeric values that shows the magnitude of reduction in drug exposure. The model predicted 41% and 49% reduction in AUC during second and third trimester, while the clinical data shows 58% reduction in both trimesters. The model predicted 27% and 34% reduction in Cmax during second and third trimester, while the clinical data from IMPACT showed 51% and 55% reduction. So this chart shows the effect of pregnancy-induced changes on AUC. Each column shows different pregnancy-induced changes. The leftmost column takes consideration of combined changes during pregnancy, and other columns only account for one different pregnancy-induced changes at a time. The ratio is measured in AUC of second and third trimester to AUC of non-pregnant. The baseline is at 1 when we did not alter any pregnancy-induced changes. So this chart shows that CYP3A4, UGT1A1, and albumin level changes during pregnancy contributed the most to pregnancy-induced changes in AUC. This chart shows the CYP3A4, UGT1A1, and albumin level changes also have the most impact in changes of Cmax during pregnancy. So the developed Bictegravir pregnancy PBPK model predicted the reduction of Bictegravir exposure during second and third trimester. The model predicted the magnitude of AUC reduction by 17% and 9% in second and third trimester respectively. And the model also predicted Cmax reduction in pregnancy by 24% and 21%. The model has some limitation because the demographic of clinical trials of pregnant population were not available. We had to conduct 10 virtual trials with 12 virtual patients in each ranging from 18 to 45 years old. And although the drug is approved as take regardless of food, there are some absorption PK changes due to food effect. With more upcoming trial design, we can expect improvement in the model. PBPK modeling is an effective tool to predict altered drug exposure during pregnancy and may facilitate clinical trial design. And further assessment are still required to determine the clinical relevance of reduced Bictegravir exposure during pregnancy. And along with pharmacokinetic study, pharmacodynamic study is still required to investigate whether the reduced drug exposure have impact on virologic response. I would like to thank everyone who are involved in the study and people living with HIV who contributed to the data for the study. And here are the list of references. And I can take any question during question session. And thank you so much for listening.